and he's going to present a very nice story about high-throughput sequencing in clinical microbiology, and he's also author of The Rough Guide to Evolution.
I am indeed, yes. It's a spare time activity.
Yeah.
It's equivalent to writing 2 PhD theses back to back in my spare time. That's what I tell my graduate students.
Well, welcome.
Thank you.
Thanks for coming.
Is it good to turn the lights down a bit, think?
Good idea, yeah.
I'll take my jacket off as well. Right, well, thank you very much for the invitation. Thank you for arranging for a car to bring me here as well, travelling in such luxury. It's great to be here in Stanford, and this is a place of legend as far as I'm concerned. It's where molecular biology was born with the with the Boyer-Cohen patents, and the home of David Relman, who was someone that I heard about when I was very young as this legendary character. Now I met him and know him. And similarly with Stan Falco, who was basically the godfather of the whole field of molecular pathogenesis. And of course, I don't think it's too far from here that Apple Macintosh was born and all those other things. So it's great to be here. So what I'm going to do is just take you through some applications of high-throughput sequencing in clinical microbiology. So in the last few years, I've actually moved a bit away from molecular pathogenesis and moved much more towards translational research, and that's what I'm going to talk about today. Look at some applications in clinical microbiology, and then look at 2 case studies and look at how the future might look. So the problem with diagnostic microbiology, particularly diagnostic bacteriology, is we're kind of stuck in a groove, a 19th century groove, if you like, in that we're using in the clinical lab microscopy and culture as the mainstays of diagnosis. And these are things that date back to the time of Koch and Pasteur. They would have been entirely familiar with the idea of looking down a microscope and growing things on solid media. And what I'm proposing is that we move fast forward into the 21st century and that high-throughput sequencing becomes a method of choice for the diagnosis of infection. And if you like, medical microbiology becomes a branch of clinical chemistry or genomic medicine. And when I give this kind of talk to clinical microbiologists, that starts to get them a bit upset. But there we are, they've got to face the future. So why should I say all this? Well, we have this new opportunity with high-throughput sequencing. This is the slide, actually, I suppose I need to update it because it says 100 times faster, 100 times cheaper. We're now probably talking thousands of times, if not tens of thousands of times faster and cheaper than conventional Sanger sequencing. So this is what economists would call a disruptive technology, something that completely changes the landscape. And Boyer and Cohen went a long way with their recombinant DNA technologies, but now with this technology we're actually abandoning having to replicate DNA in plasmids in E. coli. Everything's now done as chemistry, and we have these clonal templates, molecular colonies if you like. We've actually just published in Nature Reviews Microbiology a review, so if you're interested in anything I'm saying, I'm not going to go a lot into the technology here. If you are interested, then go and look at the review that's just come out. One of the interesting things is that there's not just one technology, there are multiple technologies all now jostling for position in the marketplace. And in a sense, we're in a kind of permanent revolution because things are getting better all the time. And if someone comes to me and says, oh, I'm writing a grant proposal, can you tell me how much it will cost to do this in year 3? I say, I have no idea. We just can't. We can project 6 months ahead, but not really any further than that. And a colleague of mine, Nick Loman, coined the term hyper-Moore's law to describe this. So Moore's law for computers, things get better every 18 months, they double in performance. Here we're probably talking doubling every year or even every 6 months for these technologies. One of the latest developments here is these benchtop sequencing platforms, which about the size of a laser printer, sit neatly on the bench and actually bring this kind of sequencing into the realm of the average laboratory, not confined to sequencing centres anymore. And this high-throughput sequencing brings all sorts of advantages. It brings open-endedness. So instead of actually devising a battery of PCRs to known targets, when you sequence the whole genome or metagenome, you're finding unknown unknowns, things you didn't even expect were to be in there. Universal applicability in that all organisms have nucleic acid. Of course, they're prions of one infectious agent that don't, but apart from that, this will cope with anything. And brings the ultimate in resolution, so single base pair changes can be detected. Now, this is the figure from our review. It's a rather busy figure, and I'm not going to go through it in detail, but just to explain the basic, this is a pointer, the basic things we go through here, the steps we go through. We start off with our template DNA, which has to be a particular quality and quantity, that has to be guaranteed. Then this is fragmented and then tagged, so that you end up with adapters at the end of your fragments. In fact, there is this new method of tagmentation where you can do the fragmentation and tagging at the same time using a transposase and various adapters. The next stage is amplification. This is done in a solid phase. Can be done either with beads in an emulsion PCR, or you can use a solid phase bridge amplification in the Illumina platform. And then down at the bottom here, you've got the various instruments that are running using these technologies, each with their own specific chemistries. Over here, PacBio, is worth stressing, actually is what we call single molecule sequencing. So you don't have any amplification here, you're actually sequencing single molecules. I just pulled this table out of the paper as well. This summarises the benchtop instruments and PacBio. And again, it's a rather busy table. I'm not going to go through it in any detail. But the key point is that there isn't an ideal technology for microbiological applications. Each technology has advantages and disadvantages. And it all depends on what you're looking for in terms of performance and throughput and read length and so forth. I'll say more about this in a moment, in a short while. Now, in the last 2, maybe 3 years, we've seen the birth of genomic epidemiology for bacteria. Virologists will say, well, they've been doing this for years, so what's so new? But obviously, our genomes are 100 times or more larger than viral genomes generally. And this has been a very interesting sea change here. I mean, one example here was the Amerithrax investigation, where they used genome sequencing there to actually identify the culture, the seed lot from which those spores came that were put into the US postal system, and actually point the finger at one individual. Here, pointing the finger at the source of the Haitian cholera outbreak. This had quite political ramifications in terms of, it turned out it was probably Nepalese peacekeepers, UN peacekeepers, that actually brought cholera into Haiti. And then there's a bit of whimsy here. If you go to the southern United States and you eat armadillos, there is a chance, genome sequencing shows, that you can catch leprosy from those armadillos. There's fairly strong evidence there that there's armadillos human transmission using that approach. This is, as I say, illustrative, not exhaustive. There are probably somewhere between 1 and 2 dozen such papers out there now. Here's a clutch that have just come out in the last few weeks. In the UK, we have this lively competition between Cambridge, Oxford and Birmingham. And Cambridge and Oxford are beating us at the moment, but we'll get our own back soon. But here's a couple of papers looking at Staph aureus, and one here looking at rapid benchtop sequencing. So, in fact, I'm going to draw on, just for the next couple of slides, on this review that's also just come out in Nature Reviews Genetics from Derek Crook's group in Oxford. And they're looking at the same question as we are. How do you transform clinical microbiology using these kind of approaches? And this is one of their figures. What they point, this is the kind of status quo ante, what it's like now. And they point out that we've got these lots of different samples that we're going to put in. Lots of different culture media have to be used. They've kind of abstracted it, but we know that there are lots of different kinds of culture media used depending on what you're looking at. You can gram stain, you can use MALDI-TOF if you've got the money to buy an instrument. Then you've got to do growth, subtyping and all this, lots of different kinds of media, lots of different kind of pipelines you're going through depending on what kind of organisms you're looking at. If you're looking at anaerobes, you have to use an anaerobic cabinet and so forth. And then you have to do your typing at the end here. And certainly in the UK situation, I don't think it's likely to be any different here. We only subtype a very small percentage of all the organisms coming through in the clinical lab. So this is a lot of effort and lots of different approaches used in the clinical lab. What they suggested, and we go along with this too, is that what you could do is you could just isolate your bacteria on agar plates and then just extract DNA, put it in the sequencer, and that would take you delivers all the same information about what species it is, what it's related to, epidemiological information, resistance, virulence, and so forth. As I'll say a bit later, we would go one step further than them, but we'll do that in a minute. Now, at this stage, it's probably, talking in a public place, I have to give a declaration of conflict of interest, and I'm going to put up a slide to show you that actually I'm not a disinterested party here. I belong to a particular sect known as Genomic fundamentalism. And this is the Wikipedia article that describes the tenets of this particular sect. So this is strict adherence to the belief that Lord Venter created the world on July the 28th, 1995. We have this creed: there shall be one diagnostic test, and that is whole genome sequencing. And we venerate various, we also believe in the imminent arrival of the sequencing singularity, which is when high-throughput sequencing becomes so cheap and so easy that it replaces everything else. We have a variety of saints here. We have Lord Rasko of Baltimore, Lord Keimer of Mary's-Rax, Lord George of WashU, the Holy Eunuch Ison, and we also have a group of old order genomic fundamentalists who actually venerate two saints who existed before Venter, Lord Stanley of Bitterroot and Lord Relman of Stanford as early prophetic forefathers. And our paramilitary or jihadi wing, or Sequence First or Just Sequence, have been engaged in the shoe leather wars, fighting shoe leather epidemiologists about which should come first, shoe leather or sequencing. Okay, so that's a Wikipedia article. Now let's get back to serious stuff. I put that because I know Stanley likes being called Lord Stanley of Bitterroot. He seems to have taken to that particular title.
Who wrote that?
I did, yeah. Yeah, you won't find it on Wikipedia. It's mocked up. Anyhow, getting back to serious things. Now I'm going to give you some case studies to root this in reality. So we start with Acinetobacter baumannii. This is a gram-negative bacillus, multi-drug resistant. We have colistin and tigecycline really as reserve agents, and there are some strains that are resistant to even those two. So we are moving towards pan-resistance with this organism. It's seen in wound infections, ventilator-associated pneumonia, bloodstream infections. We also, in the UK, and I gather it's also true in the US, have seen this in returning military personnel from Iraq and Afghanistan. So the hospital on my campus is actually where all our military personnel are repatriated when they've come from overseas conflicts. And we've seen, and you have here in the US, transmission from military to civilian patients of Acinetobacter baumannii.
How is that occurring, the transmission? Is it from
Well, because we don't have veterans hospitals. We co-locate all our military personnel going to the same hospital as the civilians in Birmingham, and so they're on the same wards, and so that's where it happens. In fact, we're hoping that it won't happen so much because we've just got a new hospital. When we used to have an old hospital that was kind of from the 19th century with big open wards, these things were very difficult to control. But we're not quite there yet. So, in terms of applications and questions, these are some of the questions that we asked ourselves at the outset, in terms of could we look at epidemiology, could we look at resistance? For this audience, I should say that we also should be looking at pathogenesis, although the pathogen biology of Acinetobacter is fairly poorly understood. We haven't actually got to grapple with that at the moment. So we looked at these questions, starting off with epidemiology. There was an outbreak in one of the old hospitals in Birmingham in 2008, and we had a number of isolates that were isolated indistinguishable by the current typing method. So the reference laboratory said, oh yes, you have an outbreak because they're all indistinguishable. And we wanted to look further than that. This has been written up as well. At the time we used the 454 platform and we sequenced 6 isolates, and then we detected SNPs, single nucleotide polymorphisms that varied among the isolates, and validated those so that they were trusted. We because we were doing this for the first time, we were very careful to go in and re-PCR out those regions, re-sequence them and everything. So what did we end up with? We ended up with these outbreak isolates distinguishable at only 3 loci in the genome, what we called SNP1, SNP2 and SNP3. We managed to polarize the changes by looking at an outgroup, and so we said this was the ancestral state and then change as you go down the table. We've got 6 patients, 4 of them are military, M1 to M4, 2 of them are civilians, C1 and C2. Now when you look at that you sort of think, well, doesn't really make much sense, can't really draw any conclusions from that. But when we overlaid that onto the time and space aspects of this outbreak, this is what we saw. So we saw these patients in an intensive care unit, 6-bedded unit, and we had 1 out in the main ITU and then 1 in the trauma unit, completely separate. What we noticed was that there was an identical genotype here in this military patient and in this civilian patient who'd been in an adjacent bed for a period of time, and where this isolate came later from that patient. So on the principle of parsimony, we concluded that this meant there had been cross-infection from the military patient to the civilian patient in the adjacent bed. You may argue, well, that's a sledgehammer to crack a nut, all that genome sequencing, just to tell you something that a shoe-leather epidemiologist would have concluded anyway. But we were quite pleased, because we had no idea whether we'd find any SNPs. We didn't know whether we may be able to make any interpretation of the data. And so even though this was only a modest step forward, we were pleased with that. It's a bit early in the morning, but some of you might have noticed that, hang on, this patient down here has got the same genotype. Well, we looked at what happened in the hospital, and it's clear that there had been no contact between that patient and the other 2. But those 2 military patients, M2 and M4, it turns out, had been repatriated from Afghanistan through the same care pathway. And our assumption is that they both got infected with this particular strain before they came into the hospital, somewhere in a field hospital or somewhere along the route. So it does make sense.
So I have a quick question.
Yeah.
How come they didn't catch the other strain from the person on the other side of the bed? The bed 5, it was also overlapped for about the same time.
Yeah, well,
there's just some strains are...
There's a strain here that didn't transmit. We don't know why it didn't transmit. Maybe that was.
Never found it anywhere else.
We didn't find this anywhere else. Well, we've only, to say this, we just looked at 6 isolates here. We've looked at much larger outbreak, which I'll talk about later. But this was just us getting started. At that time, it was very expensive, and 6 was the maximum we could afford to do. So we just looked at these ones. But you're right. You could argue that. Yeah, you're right. If you looked at this just from the point of view of, I'm an epidemiologist, I want to see what's going on, you may well have concluded that this patient transmitted to this patient rather than the other one.
I also think it's interesting that only 1 isolate made it across and wasn't like a mixture of isolates.
Yeah. Well, that's another important question. We'll come on to that in a minute. So the next thing we did was, with collaborators in London, in the Health Protection Agency and at the Royal London Hospital, we looked at some genomes from isolates from a single patient who had received tigecycline, and before therapy the isolate was sensitive. After therapy it became resistant. And so we wanted to find out what had gone on, what had accounted for that. And here we saw 18 SNPs detected between these 2 isolates, so quite a lot more than we were seeing in our previous outbreak. So we were a little bit surprised at that. But again, fortunately it made sense in that one of those SNPs was in a gene ADES, which is part of a 2-component system which is known to account for resistance in this organism. So that made sense in terms of interpretation with the current literature. But one thing we didn't anticipate was that there were some contigs in the original isolate that were not covered by reads in the second isolate. Now, normally when you think about acquisition of resistance, you think about gaining new DNA. But what seemed to have happened here is that some DNA was lost from the genome. And one of those deletions actually truncated this gene, MUT-S, which is encodes a DNA repair enzyme. And our interpretation of this is the cause of an increase in mutation rate in this particular lineage. They're still working on that to show that that is the case, but it's the most plausible explanation. And in fact, just in the last few weeks, there's a paper come out from Sharon Peacock's group working on an entirely different organism, Staph aureus, where she's seen the same thing. She's seen a lineage in which there's an increase in mutation rate associated with a loss of function mutation in MUT-S. Similar things go on in human cancer. So, I mean, there are lots of parallels in the way that these bacterial populations and populations of cancer cells evolve when we look at this kind of genomic level. Now, most recently, we've had this outbreak of what we call pulsar type 27 going over the last year or so. So, first reported case was in July last year in a military patient. And we're currently in week 60 of the outbreak. There's been 8 deaths linked to the outbreak, so it's been a considerable problem. It's been typed by our Health Protection Agency, and they gave it this unique designation of pulsar type 27. And we have now recovered 69 isolates from 29 patients, including multiple isolates from the same patient and some that were not in the outbreak. And we've genome sequenced 36 isolates from 26 of these patients so far. Now, in terms of the Schuler epidemiology, if you were just going to look at who was in the same ward at the same time, and therefore could be a plausible source of infection, this is the kind of furball that you get. And trying to make sense of this outbreak just from looking at co-location in time and space really isn't going to get you very far.
Are the numbers depicted here the order in which
they occurred in time?
Time, okay.
Yeah. So what we did was we genome sequenced. And as you can see, we're moving at a faster pace. We actually did 36 isolates now, and we're still ongoing. With this technology, we're getting 4 to 5 days turnaround time from receipt of the sample. I mean, we're not trying to race against the clock, but that just running things 2 or 3 times a week, this is the kind of figure we're getting. We identified 14 high-quality SNPs in the outbreak, and they're listed here. Again, you're not going to make much sense of them, I don't suppose, just looking at them. What we did was we tried to overlay them in terms of time and plausible epidemiological links between patients, and this is what we've come up with. Jackie Chan and Mike Halachev in my group have just put this together, in fact, just literally in the last week. One of the things that's interesting is that it's actually unclear what is the best way to display this kind of information, because there was a paper actually from Imperial College a couple of months ago showing that phylogenetic trees aren't the best way of doing it, because you have a mixture here of ancestor and descendant. When you draw a phylogenetic tree, you're assuming that everything at the bottom is descendants, and you don't know what the ancestors are. Anyway, we've got these transmission events going on. Each of these arrows represents one SNP change between a cluster, and the numbers are the numbers of the patient, and then where there's a letter after the number, that is one of several isolates from the same patient. And so you can see that there is it kind of makes sense. There is a gradual acquisition of SNPs from the earliest part of the outbreak towards the latter part of the outbreak, and sometimes we get these clusters of indistinguishable isolates that, again, these patients have all been co-located. It kind of makes sense. Just occasionally we find things where we're not quite sure what's going on. So we've seen that with these ones in black here. And in fact, we've got this, oops, that should have been coming out there. So this 3 complex, this transmission between here and 6 and 7, these latter 2 were on a different ward, but they shared communal equipment with that patient, so we're assuming that that was the transmission. And similarly, down here, patient 16 was on a different ward and tested positive after this cluster, and we think that there's some kind of transmission going on here in the plastics and the burns theatre, so environmental transmission. One patient here, patient 18, we've sampled multiple times, still ongoing. That patient still hasn't been, it's still being treated in the hospital. But you can see that we have one of the isolates from that patient in this cluster, but we have a couple of other clusters coming out of that patient. And this is a lesson that we're taking on board. When we started out, we assumed that it would just be, one patient would have everything the same, and then the next patient would have a SNP different, and the next patient would have 2 SNPs different. It's clear that that naive way of thinking is not going to to pan out. Clonal doesn't mean indistinguishable when you're looking at patient populations.
These isolates, were they obtained at different time points?
Different times and different sites as well from the same patient.
And do they
So
differ among different sites?
yes. So the first one, A, was blood culture isolate when the patient first surfaced with the bacteremia. Then there was one from a lumbar drain, two from CSF. The two from CSF, hang on. She's mislabeled this, hasn't she? That doesn't make sense. Sputum can't be D as well. Anyway, they're from different sites, and what we're learning, therefore, is that there actually is a heterogeneous population on the patient, which is not surprising when you think about it, but it's something that we're kind of learning as we try and interpret this data, and we have to bear that in mind. Again, great minds think alike. Sharon Peacock has come up with the same story with Staph aureus, and showing that she's had a patient with this MUT-S mutation who's accumulated various SNP variants within the same population on the patient.
So you've only found patients that have kind of had SNP variations where a mutation occurred in the same strain, haven't found an infection with another strain that was around in the hospital at the same time?
Not from these, not in this study so far. That is obviously something that can be done as well. And again, people in Oxford just published a paper where they showed that Clostridium difficile outbreak, supposedly, suddenly, 3 patients all with C. diff on the same ward at the same time. They genome sequenced them and found they were totally unrelated. So again, it is entirely feasible to do that. Okay, so it's going to leave Acinetobacter now and now move on to a second case study, which is German E. coli 0104, so-called Sproutbreak, named after the fact that this outbreak was linked to sprouting seeds. Initially, there were some problems with it was misassigned to cucumbers, and the finger was pointed at Spanish cucumbers, and this caused a lot of economic damage to the Spanish economy. And in fact, our German collaborators told us about some gay men who came in with saying they'd been using cucumbers for various non-eating purposes, and were they at risk and all that. But anyway, it turned out it was the sprouts. I don't know if you can misuse sprouts. 4,000, more than 4,000 cases, more than 40 deaths, in fact more than 50 deaths now. High rate of hemolytic uremic syndrome. You can see it's mostly focused around northern Germany. And females seem to be particularly at risk for reasons that are not clear. One argument might be that real men don't eat sprouts or eat salads, but that's just totally anecdotal. We really don't know what the situation is there. This just shows you the course of the outbreak, the kind of peak around the 3rd week in May, and this very large percentage in terms of the usual hit rate of hemolytic uremic syndrome associated with this sugar toxin producing E. coli. Now, right at the centre of all this was this guy here, Herr Dr. Holger Roeder, who was working in the University Clinic in Hamburg, and he was at the epicentre dealing with all these patients. He said it was just total bedlam chaos in the hospital with so many, and they had to set up sort of temporary beds, temporary wards, and so forth. And he had the isolates, and he thought, what am I going to do about this? Well, when you're in a situation like this, that any of you who follow Jerry Anderson will know exactly what you do. You call international rescue. And so he called international rescue, and Thunderbirds are go. Basically, what happened was that he managed to make a link with a Chinese postdoc, who then arranged for, not using Thunderbird 2, but using a conventional aeroplane, to take their isolate from Hamburg to BGI Shenzhen and sequence it on a new platform that had only just been launched, the Iron Torrent PGM. So BGI had one of these on trial, and they said, all right, well, we'll give it a go, and they sequenced it. Now, it so happened that I'd actually just won one of those in a European competition. So as part of their marketing, they put out this call, I spent 20 minutes filling in this competition form and actually won one. So we had one, and we were just gearing up to analyse the data. And actually what happened was that Nick Loman, who works with me, was actually at a conference at Hingston Hall on bioinformatics and public health, applied bioinformatics and public health, and he noticed that BGI released their data, their ion tolerant data, into the public domain, which was a remarkable thing. You know, people don't normally just chuck their data into the public domain like that, but these guys did. And so Nick's, having played around with some ion tolerant data already, had worked out how to assemble that, so he did an assembly and then said, well, hang on, I've done an assembly, I put it back in the public domain, how about you guys, talking mostly to the people at the rest of the conference, but actually because he did it on a blog to the whole world, why don't you come along and have a play with this data and see what we can do? And something remarkable happened then. Within 24 hours of its release, say he assembled the genome, but within 2 more days it was assembled, within a day more, it was assembled to an existing lineage by a kind of virtual MLST by a guy called Mike the Mad Biologist. What's his blog name. And within 5 days, a strain-specific diagnostic test had been released. And within a week, there were over 24 reports on the biology and evolution of the strain filed on an open source wiki. So this just is a screen dump from that wiki page, and it's perhaps not projecting as well as it should, but you can just see the remarkable international effort here. You've got people with Polish names, although he does work in England, that guy, people with Muslim names, Chinese people, Kat Holt in Australia, basically people on 3 or 4 continents all working on this. Most of these people weren't professional public health microbiologists or epidemiologists. They were just, I can use my skills here, let's have a go, and they had a play. And this, communicating through blogs, through Twitter, and so forth. Now, as it happens, I was away on vacation for the week that this kicked off. I think it kicked off in the middle of the week, about a Wednesday or so. I came back at the weekend, and I saw Nick Loman on the Monday, and he told me about all these exciting things. And I said, well, hang on, we're in the university sector here, and what we have to do is publish peer-reviewed research papers, and the pissing about with tweets and blogs and stuff like that, that's not really what we're supposed to be doing. He said, why do you have to be such a grumpy old man? And so we thought, well, what can we do? And so we contacted the Germans, and they said that, cut long story short, they said that they would welcome our help in actually writing this up and repeating all the analyses and making sure everything was sound before it went off. So we wrote it up, and we actually managed to square the circle by getting a paper in New England Journal of Medicine. Now, I've since discovered that one of the key reviewers for this paper was David Relman, so that I do owe him a bottle of champagne at some stage in the future, but that'll have to wait until he comes to Birmingham in December. What we did, we wrote this up as a journey about not just about the facts about the genome, but also about the crowdsourcing and all those things. We did have some problems because there were so many people involved, probably 24 people or so, that we kind of thought, can we put them all on? And it was a 3-way thing between us in the UK, Germany, and BGI Shenzhen. None of the 3 groups had ever met each other in real life. So it was, we didn't know what we could get away with, what we should do. And in the end, we only added 4 people from the crowdsourcing, but we then put in an author as the crowdsourcing consortium. Nick Loman repeated all the analysis to make sure that they were sound in-house. He did a heroic job in about 48 hours. And we ended up getting our paper published. In fact, it came out in the New England Journal back-to-back with another paper from Dave Rasco and colleagues where they had used the PacBio system and actually done a more extensive analysis than we had. So that was very satisfying. And another thing that I was very pleased about was that actually we listed those 4 people as collaborators, and although they don't actually appear on here, it just appears as the E. coli crowdsourcing consortium, PubMed has a special field where they put in the collaborators, so we're quite pleased about that. There was, with these kind of things, it's always very difficult to know when do you give credit, where do you stop giving credit, how do you do it, and all that. I think in the end we managed to get it right. So the takeaway messages from that outbreak is, well, infection still presents a threat, even in the most advanced societies. I mean, Germany's high up in the league tables of developed countries in terms of quality of life and all that, and here it was decimated by this outbreak. And it turns out the pathogens don't bother with passports, they move around pretty quickly. So Mike Feldgarden had pointed out that this was a strain seen in Germany 10 years before, but a similar strain had been seen in Korea. And then David Studholmer pointed out that the closest genome sequence strain was actually isolated from the Central African Republic in the late 1990s. And it belonged to an enteragrative lineage, which was completely unexpected. Everyone assumes that, EHEC, STEC equals EHEC, that they all come from these defined lineages that we know about that have type 3 secretion system, Lee-encoded type 3 secretion and so forth. This was quite different. It came from an enteragrative lineage. And that led to the conclusion that this is actually probably coming from human populations. With conventional, say, 0157, you basically look for a link to a cow pat somewhere, whereas here, that wasn't quite so obvious.
So what's the evidence that it's coming from humans?
There's no evidence that it's coming from humans, but it's just that on the balance of probabilities, rather than, oh my goodness, this must, with 0157, we always assume that it's animal feces has contaminated the human food chain.
Right.
Here, enteragrative, my understanding, and again, this is one of those things where there seems to be not a lot of consensus, is that enteragrative lineages circulate in human populations and generally are not zoonotic. I'm sure that you probably can find one or two case reports somewhere that say that's not the case, but that's my understanding, that that's the general consensus, is that they...
So like, as far as transmission of the sprout outbreak, do you think it would be a lot easier if it was an animal feces transmission instead of human waste in a developed country like Germany?
Well, the sprouts actually come from some a seed lot that came from Egypt, and they were also linked to an outbreak in France, and we don't know how they got contaminated before. I've read in the past, you see these stories about people drying out things on beaches and then people crapping on them and all this kind of stuff. You just don't know what's happened in the earlier stages of the handling of that package of food. There was this apocalyptic discussion on one blog post, which was that what we'd seen in those two outbreaks was just a small fraction of the total shipment that had come from Egypt to Europe, and that most of it had disappeared into the human food chain, and we were going to see many, many more cases surfacing over coming years. Whether that's true or not, I don't know, we'll see. It hasn't happened yet. So we saw a lot of evolution going on, so virulence factors jumping around, and pathotypes overlapping and evolving. So everyone in the field, you always like to nail everything down and have a clean, tidy kind of taxonomy in your head. But here we have a sugar toxin producing enteragrative E. coli. And I think one of the other groups who were trying to publish at the time got into a bit of an argument with New England Journal of Medicine about what term they were allowed to use. They wanted to call it enterohemorrhagic, and we said we can't call it that, and so forth. In many ways, that's a kind of argument about how many angels sit on the top of a pinhead. What you call things is somewhat arbitrary, but what is clear is that things are jumping around, and that sugar toxin phage can go into lineages that we hadn't even seen it in before. The other one worrying thing was that we saw antibiotic resistance where there was no obvious prior use of antibiotics. You don't normally treat diarrhea with antibiotics, and you're not supposed to treat STEC infection with antibiotics. But this organism came with an extended spectrum beta-lactamase encoded on one of its plasmids. So how did it pick that up? Where'd that come from? It's quite worrying. So have we actually changed anything by this open source approach? To use a couple of German words, a new Weltanschauung or a new Zeitgeist. We termed it open source genomics, and we said that it was a propitious confluence of high throughput genomics, crowdsourced analysis, liberal approach to data release. And it's clear that, although my initial impression, and lots of academics will say, oh, Twitter, waste of time, blogging, waste of time, it's clear that it can actually augment the usual channels of academic discourse. In a sense, what we're doing is we're pitting the wisdom of the crowd against the mutability of the microbial masses and who's going to win out in the future battles. You could argue we haven't really broken the mould. This kind of approach, I think, is going to be entirely appropriate when you have a public health emergency. And something similar was seen with flu among virologists. But whether ordinary science is going to suddenly change and lab books just get put onto the internet as soon as results come along, I don't know if that's going to be true. There's also this issue of site or site, as it's called, the site or site dilemma. Do you put stuff into blogs and whatever, or do you only wait till you put it onto papers? In fact, some of you may have seen that stuff about arsenic life that was published in Science. A lot of discussion there. All of the blogosphere basically ripped the paper apart. People who wrote the paper said, we're not talking to you people, we only talk through proper academic discourse channels. Have we shown the finger to Inglefinger? Inglefinger was a famous editor of the New England Journal that said that nothing that was submitted to the New England Journal could be published if it had been published in any form before. So that was forbidden. It was never raised. The New England Journal took it, even though they could see it's all been discussed on blogs and whatever. Anyway, here we are with our German collaborators having a bottle of champagne a few weeks after, and here with David Rasko as we both celebrated our New England Journal publications. Just going to quickly go through some stuff we've done since then. So we've had a paper just come out on the performance comparison of benchtop sequencing platforms. In fact, this was our plan B. When we were saying, well, we don't know if the Germans and Chinese want to play ball and we'll get a paper out, we started talking to some of our colleagues in our health protection agency and said, why don't we actually just take some isolates and sequence them on different platforms, and that will get us an interesting publication. And in fact, both plans came true. So Nick Loman drove this forward and basically compared the performance of these three different instruments, the Ion Torrent, the MiSeq and the 454 Junior, on a variety of different criteria. So in terms of cost per run, 454 Junior is very expensive compared to the others. And in terms of cost per megabase, it comes out as even more of an outlier. Ion Torrent actually are on a trajectory where they brought out a very early chip, which was really quite badly spec'd, the 314, and on that, the performance was dreadful in terms of cost per megabase because the throughput was only giving you 10 megs per run. But things improved very quickly, and they're now comparable to things like the MiSeq. Nick looked at the accuracy of these platforms, the measured versus predicted, and what he found was that the Ion Torrent was actually doing the worst of the three by some margin. It was doing better than what the Ion Torrent people predicted, but it still wasn't doing very well. And the major problem was to do with indels. So it wasn't base substitutions, but it was adding in an extra base or taking out a base at various positions in the sequence. And this is particularly the problem with homopolymers. And it's a problem with these kind of flow-based technologies. So 454 also has a problem here, but it turned out the Ion Torrent had it even worse. And so where, say, you have 8 A's in a row, the platform can't tell you whether there's 7 A's, 8 A's, 9 A's. It's just not very accurate at that. When you compare the assemblies from these different platforms, obviously you've got the two variables here. You've got the sequencing technology, and then you've got the assembly pipeline and the programs that you're using to make the assembly. So Nick tried out various combinations, and the takeaway message really is that none of them actually, we took a number of things that we thought would be of interest to clinical microbiologists. So the genes that might be used for MLST, a variety of toxins, autotransporters. And we just saw, could we recover the full length of those gene sequences using this approach from those assemblies? And in no case did we get it perfect. The best results were with MiSeq using a program called CLC, which upsets some bioinformaticians because this is not open source, it's something you have to buy. And it's a black box, nobody knows quite what algorithms it used, but it did the best of all. And then right at the bottom here, we have things like 454 Junior and Ion Torrent using Myra, giving really poor results. Only just over half of the genes actually recovered in their full length. So it's clear that, as I said at the outset, these platforms vary in their performance quite considerably. Right, the last part of the talk is what Nick Loman wants us to call shit-seek. I don't think he's come up with he's got to actually invent something for this to be an acronym of. But basically, how do we go from this I mean, Stan Falco would like this, because I know that he was. How do we go from this bread and butter of Stan Falco to sequence without culturing at all? So, going back to that earlier figure, I've made my own version of the figure from Derek Cook's paper, and basically you just take all these samples and you just shove them all in the sequencer. Well, extract DNA and then shove them in the sequencer. Why bother to culture? Takes time, takes effort, lots of different atmospheres, lots of different growth media. Just sequence the whole lot. So we did that. So we've been continuing our collaboration with our German colleagues. They have supplied us with DNA extracted from stools that they saved from the outbreak. We've had DNA from 9 stool samples, 11 runs. Actually, I didn't realise that. I suppose that's a bit of a pun, really. What that means is 11 sequencing runs rather than 11 episodes of diarrhoea on the MySeq. And we got between 1.5 gig and 3 gig of data per run, so between 10 and 20 million reads. Now this is all we only just got the MySeq a few weeks before. This is all just working out the box, no optimization. We just wanted to see what would happen. Would we get any useful information? Would it work at all? We had no idea. And interestingly, when we looked in the literature, we could only find one paper from a Japanese group where they looked at two stool samples to do this kind of thing. So it's kind of weird that nobody else has thought of doing it. Now we got some strange results. So first thing Nick did was to actually align the reads against the human genome, because we weren't interested in the human genome, and in fact we were positively disinterested in it, or uninterested in it. We wanted to get rid of it for ethical reasons. You don't want to release into the public domain, to public databases, human-related DNA sequences. And what we found, though, was rather strange, that in some of the samples, the human reads actually predominated, up to 90% or so. In others, there weren't any. So, what counts as a stool sample and what's in a stool sample obviously varies quite a lot from one sample to another. And we can't think of any artifactual reason why this might be the case in terms of what we did with the DNA. It must be something to do with the kind of samples. Nick just had a quick look at genus level assignments. He's mindful of the fact that I hate most of what goes on in microbial ecology, where they just tell you about the phylum level assignments. So clinical microbiologists, you know that there are species and genera in there, and why on earth do you keep going on about the ratio of Firmicutes to Bacteroidetes, when in fact you can actually give them proper names? So he did all that, and this is what we came up with. Sorry if I upset someone there. Or, no. And some of these I didn't know about. I mean, I'd heard of Alice Stipes because we'd come across that we're doing what we call the chicken ship project, looking at chicken gut microbiome. But Akkermansia I'd never heard of. It turns out there was a paper that just came out saying that it's actually well recognised part of the normal human microbiota. But you can see that in the ones that we thought, these are basically we were given samples that had from the conventional microbiology had Campylobacter, STEC low amounts, STEC high amounts, very high amounts, and some C. diff, and one with Salmonella. And you can see in the C. diff ones, we do actually find Clostridium here, Clostridium there. So that was kind of nice to see that was the case. Then what Nick did was to actually just say, well, let's just screen against some known virulence factors that you might say are the hallmarks of those particular pathogens, and see if we can detect those in the samples, and see if it gives us a consistent approach. And so when you look for C. diff toxin, he found it indeed in the 2 C. diff samples and nowhere else. This SLPA didn't find in one of the samples, but did in the other. When it came to looking for the sugar toxin, he found it in all of the ones that had STEC in it. When he looked for Campylobacter, he found that in the one that was labelled as Campylobacter positive. Those of you who are awake will say, hang on, what about the Salmonella? The problem is he never did find any Salmonella reads in the so-called Salmonella sample. We don't know whether that's because we were given the wrong sample, or Salmonella was there in very low amounts, or quite what. This is all hot off the press, so we haven't got into the details of that yet. when he actually then did, what he then did was to actually look at the reads and align them against the reference, because we knew what this German STEC looked like in terms of its genome. So he just aligned them and looked at the depth of coverage in each of the samples. And so this is basically going along the genome in terms of coordinates along the genome, and this is the amount of coverage that he got. So you can see he's got, in this sample, which is so-called STEC very high, he got about 20-fold coverage of the genome in the sample. Again, we were very pleased with this because we just did the experiment. There was no optimization or anything like that. And one interesting thing was there's this little upswing here corresponding to the sugar toxin encoding phage. And that goes with the biology of what we know about the infection, which is that the sugar toxin is actually released during the lysis induced by the phage, and that during that lysis, obviously the phage is making many, many more copies of its genome than compared to the host. So that was nice. That made sense. There's another one where it got, this doesn't quite make sense because this was said to be STEC low, but actually gave much higher coverage, 200-fold. You can tell we did the experiments rather than made the results up because they don't always come to what you expect. But again, we saw this nice little upswing where the sugar toxin encoding phage was in the genome. But here's another one that's supposed to be high, and in fact we only got 5-fold coverage of the genome, but we did not see any upswing there associated with the phage genome. So, I mean, it's hinting at the fact we might be able to uncover new things about pathogenesis of infection by looking at things in this way, because what's going on here? Was this patient in a different phase of their illness? Or was this a different kind of sample that had been taken in a different way? It's unclear, but the facts are there that we don't see the phage overrepresented as we do in all the others. And then just to show again one where we did get this supposedly high amounts of STEC, but in fact the coverage was still only 5-fold, but here clearly we did see the phage. So it wasn't an artifact of our pipelines or anything like that, because two samples behaved quite differently. Campylobacter, this one had 90% human DNA, and basically he could only detect these toxins associated with Campylobacter at very low levels. And then it's about 5-fold coverage across. Wait a minute. Yes, it's 5-fold coverage against 0104. Now, what does that mean? Does it actually mean that it's got 0104, or does it mean that it's actually got another E. coli in there, and we're seeing some of that coverage, and that's not been unravelled yet? And this is the issue: is that even in a normal person, you're going to see E. coli, and how do you work out whether they've got, most of your reads are coming from one source or another, and that's something we're still struggling with. And then Clostridium difficile, again, he managed to detect at very low levels the toxin A and B, but if you actually look at the percentage of each taxon, C. difficile doesn't even figure in this list of top 10. It's down at sort of a fraction of a percent. So we're still finding our way with this. Anyway, I better shut up now. So basically, genome sequencing brings all these wonderful opportunities: open-endedness, universal applicability, resolution. Benchtop sequencing is poised to revolutionise things. The platforms have defined strengths and weaknesses. Also worth saying that you ain't seen nothing yet, because although these platforms are getting better, there is another platform waiting in the wings that may change the landscape yet again. That's called from Oxford Nanopore. Now, there was an announcement at a conference earlier in the year where they said they had a product. They basically had a USB stick like this, which you pipette your DNA. The DNA goes through this pore and is read. No sample prep, no library prep necessary, and the data just streams through the USB port into your computer. That's what they claim, and this is going to cost a few $100s. Now, since they made that announcement, nobody has seen any of these instruments. They've not released them. They've not even released into the public domain data produced from this platform. So there is, is it hope or is it hype? We don't know. We're all waiting to see when they're actually going to have something that we can all get our hands on and try it out. It has to be said, with the iron torrent, the hype that went before wasn't matched by the reality when it first came out. They actually released it at rather less spec than we expected. So we don't know, but this will transform the landscape again. all this issue about trying to assemble short reads into contigs and scaffolds, that will go away if you can get 10,000 base pairs in a single read off of an instrument like this for a few $100s. Now, I am a genomic fundamentalist, but I do actually have my doubts sometimes in the early hours of the morning. Genotype doesn't always predict phenotype, does it? It can be hard. A single base pair change can make something resistant or make something not pathogenic when it was, or vice versa. So there will be a lot of things that we can capture easily from the genome, but there will be some things that won't be so obvious, and we have to just be humble. But we don't know how far we can push this until we actually try. The other problem is this heterogeneity in bacterial populations, and we really have to sort of get to grips with that and work out what it means, both in terms of understanding the epidemiology and understanding biology. And in fact, the heterogeneity was actually the key to the Amerithrax investigation. What they did there was when they plated out, they saw colonial variants at very low numbers, less than 1 in 50. If you'd have just genome sequenced the whole population, you wouldn't have seen that. It relied on people actually saying, oh, that looks different, and then picking that colony and then sequencing it. And so we have to be careful about being overconfident. And sometimes indistinguishable really does mean identical. We don't know what the rate of change is in each lineage until we actually go out and measure it. But we know that in certain, if you tried to track the spread of leprosy through Mumbai, let's say, you wouldn't be able to use SNPs because that organism grows so slowly and acquires mutations so slowly that you could go into Mumbai and sample every M. lepri genome and they'd all be the same. It wouldn't tell you anything. That's at one extreme. The other problem is that clinical microbiology is a very conservative discipline, and I live in an ivory tower, you might argue. I mean, luckily my ivory tower is on the same campus as a real hospital, and we do talk to the people in the real hospital, but there is an issue about how do we actually move this into a hospital environment. How do we communicate with clinicians? They stumble over long names like Stanotrophomonas. That kind of freaks them out when you use a bit of kind of Latin and Greek on them. If you started then giving a long multi-page report telling them all about the SNP types and the virulence factors and the resistance genes, how's it going to work out? The other point is that, although I've kind of egged up a bit saying that we're going to just sweep away everything that's gone before, sometimes that doesn't happen. Radio, you might argue, is an inferior technology to TV, but nonetheless people still use radio. When you're in the car, driving on the freeway, you're going to listen to the radio. You wouldn't watch television. And it's quite likely that culture will still be required in certain parts of clinical microbiology. And this sequencing will augment what we do already. It may replace some of it, but may not replace all of it. So, in short, the promised land is in sight. Is Moses looking at the promised land with all these sequences shimmering before him? But there are still many rivers to cross before we actually get there. But we've started on that journey at least. And that's me finished. I just put all the acknowledgements up. Oops, there we are. That's the right acknowledgement slide. I've got 2 acknowledgement slides. A lot of the work here has been relied on Nick Loman, lots of other people as well. The Acinetobacter work, Jackie Chan, Mike Halachev, Pallen, Oppenheim, particularly need to be mentioned there. And if you're interested in this kind of stuff, we do a blog, which we keep up to date occasionally. But we put interesting stuff. And myself and Nick are both on Twitter, and I'm recording this talk and I'll put it up onto YouTube later. I'll put all my talks up onto YouTube if you want to hear any of them further. And that's it. Thank you. Hello?
I completely agree with you that sequencing is extremely powerful in diagnostics. Where do you see this going, that eventually each lab, and there I mean academic lab as well as clinical lab, has their own sequencing machine, or will there be sequencing centers where people just submit their samples?
Well,
Also thinking in terms of the different technologies that may be complementing each other.
it's very hard to predict. I mean, one view is that if we're going down the route of benchtop and then we're going to go to USB stick sequencing, let's say, every lab can just do its own sequencing, and that's, what's the issue? It's going to be straightforward. Some people have said, well, you can do that and then you'll have to have a big database to compare what you've got like we have MLST databases now, you'll have a genomic epidemiology database that has 20,000 or 50,000 or 100,000 or million Staph aureus genomes, and you can slot yours in and say that genotype was last seen in a hospital 50 miles away, or was last seen somewhere the other side of the world. It must have come off an aeroplane. It's very hard to tell. I mean, the argument in clinical microbiology in the UK is how far should you consolidate labs and have, say, in our city of Birmingham, where we've got 1 million people, should we have one big microbiology lab and lots of motorbikes taking all the samples there, and then you get the economies of scale in processing everything? Or do you keep the labs actually close to the patients in the hospital so that interactivity is there? And I don't think there's it's not a self-evident correct answer to that. It's just how the marketplace plays out with convenience and how these technologies are going to evolve. I mean, as automation is kicking in even in the clinical microbiology lab, I think the arguments for centralisation do become stronger. But we'll see.
I was curious about the poop samples that you sequence because the high variability of human DNA present in them, and I'm thinking of how people usually diagnose UTIs looking at human epithelial cells. So do you think that reflects something in the course of when they were, like, was the diarrhea worse in those people? Have you been able to correlate them to any patient?
We haven't, because when we started looking, we thought, well, maybe we could take receipt of the samples, and then we thought, no, we don't want to fly STEC containing feces across international borders. It's going to create an awful lot of safety problems and customs and import. You know, what have you got in your bag? I've got some highly infectious STEC. Yeah. So we said, well, we'll just take receipt of the DNA. So that has meant that we haven't actually got that ability to go and just look and see, well, did it look watery or did it look, you know,
But
kind of...
presumably when they collected it, the physicians, I mean, or even just the...
Well, they may be able to thaw it out and say, yeah, that one was just looked like rice water stool, and that one looked like a normal feces, but just a little bit, you know, less formed than usual or whatever. But again, it's one of those things. I don't think anyone's done this before, and you sort of think the Human Microbiome Project, you thought they would have done that, but we haven't found anywhere where they've actually done something like this, where you've taken different kinds of diarrhea. I mean, there's this whole issue of, on one case we did a technical replica, and we did achieve fairly consistent results. But there's this idea of biological reproducibility. If you're taking stool samples, and it's not a nice thing to think about, how do you actually collect a stool sample and the ways in which it's done. You know, how much variation is there? We don't know. I mean, I was taught way back when that human feces is 1/3 bacteria, 1/3 sloughed off intestine, and 1/3 remains of your food. So if it's really 1/3 sloughed off intestine, then it's surprising we're not seeing more human DNA than we are. So you know, it's interesting. We're in kind of terra incognita really with this.
How do you see, I don't know if you guys plan on doing some more of this work or just other groups taking a lot of this really great sequencing data from all these different outbreak strains or just hospital cases and connecting to that to the pathogenesis of the disease or anything to other, you know, connecting some of those phenotypes or virulence factors?
Well, that's that last slide, the caveats about actually trying to predict virulence from a genome is a hard ask. I mean, it's something that so with this particular outbreak strain, why did this outbreak strain decimate Germany like that? What was it about it? Some people have said, well, maybe it's because it's got these particular adhesins. Maybe that's the source of it. I mean, we're all surprised. I mean, we all just work on kind of handy myths. I always thought, you know, pathogenesis of hemolytic uremic syndrome required that you have a Lee-encoded type 3 secretion system to mediate the initial adherence to the bowel and all that. Doesn't seem to be the case. So, you know, in a broad sense, finding the kinds of adhesins, the kinds of toxins and other things that you kind of recognise will obviously inform your thinking. But often you'll find things hidden in the genome you didn't even recognise were virulence factors, and it's very hard to tease it out. And even when you do recognise them as virulence factors, genome sequencing is quick and easy. What Nick disparagingly calls experimentomics. are actually going and doing experiments and hypothesis-driven research to show what you've seen in that genome really is what you think it is. That takes months or years of postdoc and graduate student time to work out. So that's the issue, is that we can collect lots of sequence data. But we're finding new things. That's the fact that some of those stool samples we could see the phage and others we couldn't provokes new questions. What's going on? We thought lysis was part of the pathogenesis. Why don't we see it? I mean, we need to collect more samples. I mean, metatranscriptomics is even the next stage on, which genes are actually being expressed. We haven't tried that yet. Okay.
Great, thank you very much.
Well, thank you. Thank you for listening.